All right, good afternoon, everyone. Can everyone hear me? Okay. All right. Well, um, so just a few words on the exam from last week. So uh, I don't know what y'all are doing, but just keep it up because the overall average on the third exam from last week is an 86, which I don't think I've ever seen before. Um, there were a total of 19 A's, 8 B's, 2 C's, 1D, and only 3 F's. I mean, that is extraordinary. I've never seen grades like that. So y'all are doing extremely well. So just keep it up, whatever you're doing, and give yourselves a pat on the back or whatever. But you're doing extremely. So congratulations. Yeah, right. Yeah, thank you. But y'all are doing all the work. So y'all are the ones that are studying, and I'm just sitting here blabbing for an hour and a half. So anyway. So today we're going to get started on something totally different. We're going to talk about antibiotics today. Now, um, an antibiotic is we've kind of touched on antibiotics a little bit before, but we haven't really gone into any detail. If we had to define what an antibiotic is, it's basically this. Okay, this slide here kind of sums up what an antibiotic is. It's basically a chemical that either inhibits the growth of or kills bacteria. Now, most antibiotics, at least in their like original form that they were found in, um, were made either by a bacterium or bacteria and fungal cells, fungal organisms. Um, Now there are some synthetic versions that were made in the lab. We'll definitely talk about those. But if you think about the original source of some of the original antibiotics that were discovered, it was either made by a bacterial cell or a fungus. In fact, you might remember that our very first antibiotic that was ever discovered was penicillin. And does anybody remember who discovered penicillin? What was the name of the scientist? That's correct. It was Alexander Fleming. And it was something that he wasn't looking for. He discovered it totally by accident. He was growing these plates of some bacteria, and he pulled his plates out of the incubator, and he noticed that there was mold growing in his plate, like that. And he thought he had all this mold contamination, which he did. But right as he was about to throw away all of his plates, he noticed that right around the mold, there was no bacteria. He noticed what we now call a zone of inhibition right around the mold. And so after he did a lot of other research, he was able to isolate what we now know as penicillin. Okay. Um, so again, penicillin. Now again, if we look at some of these other antibiotics here, you can see that most antibiotics that are used to treat a lot of infections were either made by a mold or a bacteria. So, for example, the penicillins and cephalosporins, those are made by molds. And then the next, five here, streptomycin, vancomycin, erythromycin, tetracycline, and bacitracin, those are all made by some type of bacteria, especially streptomycin, vancomycin, erythromycin, and tetracyclines. Those are all made by Streptomyces species. But bacitracin, which you probably recognize, was made by a bacteria, is made by a bacteria called Bacillus subtilis. Okay, and there are some synthetic versions that were totally made in the lab, and there are even drugs that we call semi-synthetic versions. So, for example, and we'll get into this later on, there are what we call some semi-synthetic versions of penicillin, where through overuse of penicillin since the day that it was first discovered to today, most bacteria are resistant against penicillin. They've developed some sort of resistance against it. And what a lot of the drug companies have had to do is to modify it somehow to make it work. And so one of the things that they do to modify these drugs, can you guys hear me? I have a comment here that y'all can't hear me. So again, apparently my sound is coming through. So if you're having issues hearing me, just log out and log back in. Usually that resolves the issue. All right. But again, coming back to this, what we call a semi-synthetic version is you take the original version of the drug that was made by the mold or the bacteria, and you modify it a little bit. You add a chemical group here to make it work again. And we'll talk a little bit more about how we do that in a little bit. All right. Now, again, before we get into any specific antibiotics here, I want to define some other terms here. The next thing I want to talk about is what's known as the chemotherapeutic index. A chemotherapeutic index is where you have to find a balance between the amount of the antibiotic that can cure the infection and not at the same time harm the patient. Because the idea here is like, okay, if a person has a raging infection inside of them, I could pump them full of a really high dose of antibiotic, and I would probably cure the infection. But the problem is that really, really high dose of that antibiotic would probably be so toxic to the patient that I might end up making the patient sick and killing them. So I can't just give them a really, really high dose and say, okay, we're going to cure the infection. So I've got to find a balance between what is the amount that's tolerated by the patient and the minimum that can cure the patient. And so I have to find what's called the chemotherapeutic index, which is you take the maximum dose that can be tolerated by the patient and not get sick, and you divide that by the minimum dose that can cure the infection, right? And when you find that balance, what you found is a drug that is selectively toxic, okay? What this basically means is a drug that is selectively toxic is one that targets the microbe causing the infection and not the host, okay? And that's what we want. We want a drug that has that is selectively toxic and targets the pathogen, the microbe causing the infection, and not the host. And an antibiotic that I would call a good antibiotic is one that has a high chemotherapeutic index. That means that it's being selectively toxic. It targets the microbe and not the patient. It's not very toxic to the patient. Okay. If I say that a certain antibiotic is a quote unquote bad antibiotic, that means that it's not selectively toxic. It would have a very low chemotherapeutic index, meaning that it has more of toxic effects on the host's cells and not much to the pathogen causing the infection. So long story short is we want an antibiotic to have a high chemotherapeutic index, targets the microbe and not the host. Okay. Now, some other terminology that we need to get through here. Some antibiotics are referred to as either being broad spectrum or narrow spectrum. A broad spectrum antibiotic can kill a wide variety of different organisms. So for example, if you have an antibiotic that can kill a lot of gram positives and a lot of gram negatives at the same time, then it would be a broad spectrum. But if I have an antibiotic that targets, let's say, a very specific group of gram positive bacteria, or a very specific group of gram negative bacteria, or maybe even one single type of bacteria, then it would be considered to be a narrow spectrum, right? Now, broad spectrum and narrow spectrum antibiotics are given all the time. If you go to your doctor, or if somebody goes to their doctor and their doctor prescribes them a broad spectrum antibiotic, what does that mean? What do you think that means if they're being given a broad spectrum antibiotic? Right, but why is the doctor giving a person, I guess I should have rephrased, why is the doctor giving the broad spectrum antibiotic rather than narrow spectrum? think I saw the answer here. Yeah, so basically the reason that they would be doing that is because the doctor doesn't know what the person's infected with. So to cover all the different possibilities, they would give a broad spectrum antibiotic. Okay, so broad spectrum antibiotics, they definitely have an advantage, because they can kill a wide variety of different organisms. But that's also a disadvantage. What is one of the problems that you could think of by a person being given a broad spectrum antibiotic? That's correct. It can kill your normal flora. So with long-term use, broad spectrum antibiotics can kill your normal flora bacteria, which then can make you susceptible to being infected by more harmful things. So you kind of have to watch out for that. All right. Now, other antibiotics, they can either be described as being bactericidal or bacteriostatic. Bactericidal antibiotics, they kill bacteria. Whereas bacteriostatics, they don't. They just inhibit, they inhibit growth of bacteria. All right. As far as how we administer antibiotics, if it's an antibiotic that can survive the acids in the stomach, it might be given orally. For a lot of infections on the skin, like wound infections or other things, you might be given a topical cream or a topical ointment. But some antibiotics have got to penetrate tissues to get really into and treat systemic infections. And when that happens, the only way to really administer the drug is through what's called the parenteral route. Does anybody remember what we mean by the parenteral route of administration? What is that referring to? That's correct, like an injection. So maybe through IV fluids or an injection, exactly. All right. Now, we're going to go through a lot of different antibiotics here, a lot of classes of antibiotics. And obviously, there's a lot to memorize or learn here. And so what I tried to do was kind of distill down, out of all the different things you need to know, what are the three most important things? Kind of going in order from the most important to, I wouldn't say least important, but kind of making a list here. Let's put it that way. So as you're studying these different antibiotics, the number one thing that you need to know about all those different antibiotics is, how do these antibiotics work? What is their mode of action? In other words, when that antibiotic gets into a bacterial cell, what is it targeting? Okay, is it targeting the cell wall? Is it targeting the ribosome? What is the actual target of the antibiotic? The mode of action. So that is the number one thing that you should know. Okay, if you look in your lecture notes, the charts listing all of the antibiotics, they're actually organized by their mode of action. Okay, to kind of make it easier for you to break down. So mode of action. Second on the list, as far as the things you need to know, is have a general idea of what types of organisms these antibiotics can be used to treat. Now, some antibiotics, they can kind of be really used to treat a lot of different things. So it's kind of hard to really say, to kind of really remember them. But one of the things that you want to pay attention to as I'm lecturing is, if I'm talking about some antibiotics and I say, okay, this antibiotic is really generally used to treat this, this, and this. And if I do that, and I'll kind of highlight those areas there. If I say that a certain antibiotic is usually used to treat this and this, then you should definitely know it. Okay. Or if I say that there's a certain antibiotic that's a really, really broad spectrum antibiotic, you should know that. Okay. So I'll kind of just really pay attention to, as I'm lecturing, kind of focus on that if I say that this is for this and this is for this. Okay. And then some antibiotics, unfortunately, have some side effects or toxicities. And as I'm going through any of these antibiotics, if I mention antibiotics that do have any side effects or toxicities, you should know those. Okay. But again, even there's a list of things here, the number one thing that should be at the very top of your list is definitely know the mode of action. Okay. Know how the drug works. Okay. Now, here are some of the different modes of actions for antibiotics. the first thing that we're going to get into is to talk about antibiotics that inhibit the cell wall. All right. Then we're going to talk about some that disrupt or break down the cell membrane somehow. Then we're going to talk about some inhibitors of protein synthesis. And those antibiotics, they do so by usually targeting the ribosome. Then we're going to talk about some antibiotics that interfere with a really important metabolic pathway. And then finally, with antibiotics, we'll talk about some ones that inhibit DNA synthesis or RNA synthesis. Okay. So again, these antibiotics, when I'm using the term antibiotic, we're really going to focus on these chemicals that are really used to either kill or stop the growth of bacteria. Now later on, and we probably won't get to this today, but later on we'll talk about antiviral medications and antiprotozoan medications, but that's not what we're talking about right now. Right now we're talking about antibacterial medications, okay? And these are the different modes of actions for these things. All right. So what we want to focus on first are our first really important mode of action, which is the inhibition of the cell wall. Okay, now, before I get into specifics here, when we think of cell wall synthesis inhibitors, cell wall synthesis inhibitors can be basically broken down into two main groups. We have what are a class of drugs that are called the beta-lactam antibiotics. And then we have what are called the non-beta-lactam antibiotics. I mean, they both classes they inhibit the cell wall, but they do so in a little bit of a different way. Okay, and structurally or molecularly, in terms of their structure, they look a little bit different. Okay, so we have the beta-lactam antibiotics and the non-beta-lactam antibiotics. We're going to start off by talking about the beta-lactam antibiotics. And these beta-lactam antibiotics, they have a very specific target in the cell wall. Remember when we talked about the cell wall of bacteria, we said that the cell wall of bacteria is made up of a peptidoglycan, right? So the peptido part means that there's protein in there, and then the glycan means that there's a sugar component to the beta-lactam. So just kind of really quickly going through this here, remember that the sugar part for the cell wall is made of these alternating NAG and NAM sugars. So you basically take a NAG and you alternate it with the NAM. NAG, NAM, like this. All right, so that right there, that would be what I would call a sugar chain of NAGs and NAMs. And what bacterial cells do is they build a bunch of these lying parallel to each other. Okay. All right, but then these NAG and NAM sugar chains, they're bonded to each other using a peptide crosslink. So this is the protein part of the peptidoglycan. So the beta-lactam drugs, what they do is they inhibit the synthesis of the peptide crosslinks. So they basically tell these bacterial cells, you cannot make peptide crosslinks anymore. We're going to block your ability to do that. Well, the problem with that is that now these bacteria that have been exposed to these different beta-lactam drugs that we're going to talk about, their cell walls are not very rigid anymore. And when this happens, they start absorbing a lot of fluids and salts. And so all these different fluids and salts rush into the cell, and that'll cause the bacterial cell to swell, and then pop by lysis, which normally, if they have a cell wall that's intact, they can't do that. Okay, so these beta-lactam drugs, they all kind of work the same way. They inhibit the peptide cross-link production. All right, so let's look at a couple of these here, or a few of these. All right. So the first, as I said, group of cell wall synthesis inhibitors that we want to take a look at are the ones that are called the beta-lactams. And they include penicillin. They also include some synthetic and semi-synthetic versions of penicillin. They also include another group of antibiotics that are made by molds called the cephalosporins, and also what we call the carbapinams. So all of these are beta-lactam cell wall synthesis inhibitors. They're obviously different drugs, but they all have something in common. Okay, what they all have in common is something that's referred to as the beta-lactam ring, which in this diagram here is this part of the drug. Okay, this is called the beta-lactam ring. This beta-lactam ring is extremely important because it's the active part of the drug. In other words, this is the part of the drug that makes it work. Okay, this is the part of the drug that allows these antibiotics to inhibit the peptide crosslinks. All right. Without this part of the drug, without the beta-lactam ring, these drugs are ineffective. Okay, which will lead us into something that we'll talk about later on. Okay. One of the ways that these drugs are different is what's attached right here, where it says R. Okay. Usually, there's some different chemical group that's attached there that differentiates between these different antibiotics. But regardless, they all have this part. They all have that beta-lactam ring, which is, again, the active part of the drug. So with penicillin being a beta-lactam drug, it works the same way. And, penicillin, there are some advantages to it and some disadvantages to it. Again, penicillin was the very first antibiotic that was discovered. Let me see here. What was that? Shanette, the other part of the drug are ID markers. No, the other parts of the drugs are just different chemical groups that have some other function for the antibiotic. And but the main part of the drug is really that beta-lactam ring. So that's the one that you want to focus on the most. Okay. So with penicillin, like I said, it back 40, 50 years ago when it was first discovered, or probably going way beyond, even 60 years ago now, when it was first discovered, it was a really good antibiotic. It was used to treat a lot of different major and minor infections. But the problem is that since penicillin was overused so much, we've gotten to a point now where most bacteria these days are resistant against it. It just doesn't work. We'll talk about what bacteria that are resistant make to become resistant. But the point is that they don't work anymore. And so a disadvantage is that through overuse of these antibiotics, not just this one, but a lot of antibiotics, resistance develops. Okay, and you have to kind of discover or make new versions of them. Another disadvantage is that some people, as you know, and some of you out there also might experience this, are allergic to penicillin. And this doesn't happen in everybody, but in some individuals, penicillin is a we talked about haptens in the immunology section, where a hapten is a molecule that is usually a very, very small molecule that doesn't really elicit an immune response by itself. But in some people, okay, it can combine with proteins in the body and become something larger that your body can then recognize. Okay, and in those individuals that are allergic to penicillin, it does that. So there's some disadvantages to it. So what we want to talk about is with something like penicillin, if you have bacteria that are resistant against penicillin, what is it about these bacteria that make them resistant, and what can we do about it? How can we modify these antibiotics to make the antibiotic work? Okay, well, let's first talk about what makes an antibiotic resistant to a beta-lactam drug like penicillin. And what that is where bacteria that are resistant against these beta-lactam drugs, they produce this enzyme that some of you may have heard of before called beta-lactamase. Some certain textbooks will refer to them as penicillinases. I prefer the word beta-lactamase just personally, but um, if a bacteria produces this enzyme or has the ability to produce this enzyme, what they're doing is they're using that enzyme to destroy the beta-lactam ring. And remember that I said that the beta-lactam ring, that is the important part of the drug. That's the part of the drug that makes the drug work. So without that part of the drug doesn't work and the bacteria survive. Okay. So again, with something like penicillin, the penicillin in its natural form, as it was discovered by Alexander Fleming, it pretty much doesn't work anymore. I mean, there are some exceptions to that, but most bacteria are resistant against it. So what manufacturers of a lot of these antibiotics have had to do is they've had to modify penicillin. They've had to add on a chemical to it that makes it work. So that kind of brings me back to this part of the drug here, right there where it says R. Okay. A lot of these beta-lactam drugs are different in terms of what's attached right here. Now, what they do, to try to put it simply, is they add on a chemical group there that has a similar structure or looks like the beta-lactam ring. It's not the same, but it kind of mimics it. And the idea here is that when the antibiotic is exposed to the beta-lactamase enzyme, that the beta-lactamase enzyme attacks whatever we attach here and leaves the beta-lactam ring intact. And if we can do that, then the drug will still work. Okay, so it's like trying to preoccupy, or we're trying to divert the enzyme to attack whatever we attach here and say, leave this alone. Okay, let me give you some examples of some semi-synthetic versions of penicillin. I'm not going to go through the whole list here, but methicillin, for example, ampicillin, amoxicillin. There's a lot of these antibiotics you've probably heard of before. All of them are beta-lactam drugs. They all have the beta-lactam ring, but they've been slightly modified from compared to the original penicillin version of them. And what the drug manufacturers have done is they've added on these different chemical groups. Now, these different chemical groups, if you pay attention to what they look like, they have a ring structure. Okay, they don't look exactly like the beta-lactam ring, but they are similar. They're similar to it, so that if they're exposed to these beta-lactamase enzymes that some of these resistant bacteria make, the hope is that the enzyme will attack this part of the drug and leave the beta-lactam ring alone. Okay, and that if that happens, then the drug will still work. Okay, but again, what happens usually is that a lot of these antibiotics, they're just overused so much that these drugs might work for a little while, and then you start evolving a whole group of bacteria that are resistant against those, and you have to have these drug manufacturers. They constantly have to tinker with the antibiotics and play with them and add on chemical groups to continue to make them to work, to have them to work. Okay. So kind of running through this list here, we'll talk about methicillin and ampicillin later on, but like amoxicillin, right? I'm sure most of you have heard of amoxicillin. It's a very, very common antibiotic. If you have ever had an ear infection, or if you have kids and they've had ear infections, my son right now, he has an ear infection and he's been on amoxicillin for the past 9 days. It's one of the more common antibiotics given for to treat middle ear infections. Well, amoxicillin is basically a modified version of penicillin. It's where they take penicillin and they add on this chemical group to make it work. Well, there's actually a more powerful version of amoxicillin. Some bacteria have developed resistances against amoxicillin. So what they've done is they've taken amoxicillin, which is already a modified version of penicillin. They played with it a little bit more. They modified it, and they've created this drug that's called Augmentin, which again, I'm sure some of you have also heard of. Augmentin is basically amoxicillin with this chemical attached to it called clavulanic acid. And so clavulanic acid is just another chemical that has been attached to this to preoccupy the beta-lactamase enzyme to prevent the drug from being destroyed by it. So again, every time these bacteria develop resistances against these beta-lactam drugs, we modify them so that they can work better. Now, these are, again, what we refer to as these synthetic or semi-synthetic versions of these drugs. Now, some consequences of creating these synthetic versions or semi-synthetic versions of beta-lactam drugs. Well, for one, they're typically broader spectrum. Penicillin, if we go back to the original version of penicillin, for the most part, only works against gram-positives. Now, there are some exceptions to that. There are some gram-negatives that do respond against penicillin, but for the most part, it's usually good only against gram-positives. But with something like ampicillin or amoxicillin or certainly Augmentin, those are semi-synthetic versions of penicillin, and they are broader spectrum. They can be used to treat gram-positives and gram-negatives. So that's one of the main consequences of creating these different versions of these drugs. Now, another group of beta-lactam drugs that are similar but are used against things through the different things are the cephalosporins. So cephalosporins, they are also produced by a mold. They're produced by the cephalosporian species of molds. And they seem to have some more resistance against being destroyed by these beta-lactamase enzymes. And currently, there are like 3 or 4 different generations of the cephalosporins that have been discovered or developed over the years. Years ago, when cephalosporins were first discovered, the very first group of cephalosporins that were first discovered were called the first generation cephalosporins. And the first generation cephalosporins were fairly narrow spectrum. They were only really good against gram-positives. And they were used for a while and to treat certain types of infections. But then through overuse, what happened is that bacteria developed resistances against the first generation cephalosporins. So the manufacturers of these antibiotics, they tinkered around with the antibiotic, they played with it, and they modified it, and they created these second generation cephalosporins. Now, the second generation sporins compared to the first are a little bit more broader spectrum. Not only can they be used to kill gram-positives, but they can also be used to kill some gram-negatives. And so these second generation cephalosporins were used to treat, infections, and they worked for a little while until those bacteria developed resistances. And so they had to modify the drug again, and they created these third generation cephalosporins, which are even more broader spectrum. They can be used to kill even more gram-positives and even more gram-negatives. The point here is that with every generation of these cephalosporins that were produced, the antibiotic became more and more broader spectrum. All right. Now, here's some examples of some cephalosporins, some first, second, and third generations. Now, you don't need to know the brand names. Just in general, know what we mean by first and second and third generation. So first generation cephalosporins like Duracef were primarily used against gram-positives. Second generation cephalosporins like C-chlor were used against gram-negatives and some gram-positives. And third generation cephalosporins are even broader spectrum. They're used against even more gram-negatives and even more gram-positives. In fact, if a person has an infection by Pseudomonas, the antibiotic that's usually preferred to kill Pseudomonas infection is a third generation cephalosporin. Okay, so again, they get broader and broader spectrum. Now, another type of beta-lactam drug are a group of beta-lactams that are called the carbapinams. Carbapinams, they are extremely broad spectrum antibiotics. And they're not given all the time. They're usually given as a last resort, where if a patient was given a lot of other antibiotics and none of those worked and the patient's going to die, and you got to save the patient, then maybe these carbaponyms will be given. It's just that carbaponyms are so strong that they're so extremely broad spectrum that they can kill off all your normal flora bacteria, making you very, very susceptible to other infections that you normally wouldn't get infected by. So they're typically given as a last resort. Now, even though these antibiotics are very powerful, the problem is that we're starting to see more and more bacteria become resistant against carbaponyms. I don't want to go into all that, but if you have a group of bacteria that are resistant against the carbaponyms, they are almost impossible to treat. They're like truly, truly, truly super bugs. They are just very, very, they'll almost always cause people to die from whatever that infection they're causing. Okay, so all of these cell wall synthesis inhibitors we've talked about thus far were all of the beta-lactam drugs. But we also have non-beta-lactam inhibitors of cell wall synthesis. Now, before I talk about them, if we go back to this diagram that I drew, if it loads up. So remember that I said that the beta-lactam drugs, what they do is they target the peptide crosslinks, right? Well, the non-beta-lactam drugs, what they target is they target the bond between the NAG and the NAM within a sugar change. So the non-beta-lactams. So they target between the NAG and the NAM within a sugar chain, and if that is disassembled, then you basically are disassembling the entire cell wall. Okay, so the non-beta-lactam drugs, that's how they work. So there's two that I want to talk about here. Go back to my slide. One of these is something that I'm sure you've heard of before, bacitracin. If you have a tube of Neosporin at home or something equivalent to that, it's generally used as a topical ointment for skin or wound infections in the skin. And one of the antibiotics in there, it's not the only one, but one of the antibiotics in there is bacitracin. Bacitracin is produced by a bacteria called Bacillus subtilis, which is a very common bacteria that's found everywhere. But it's fairly narrow spectrum. It is primarily used against Staphylococcus infections and Streptococcus infections, which, if you think about it, in the skin, if you have a wound in the skin, most likely, compared to other bacteria, most likely that wound would be more likely to be infected by either some sort of Staphylococcus or Streptococcus bacteria. So putting a little Neosporin on there would prevent infections by those. And then another non-beta-lactam antibiotic is vancomycin. Vancomycin is a fairly new antibiotic that was developed to fight MRSA infections. So, okay, just to kind of give you a bit of a backstory here. A long time ago, I mean, I'm talking like 50, 60 years ago, if a person was infected with Staph aureus, they would most likely have been given penicillin to treat that Staph aureus infection on the skin. But what happened is that through overuse of penicillin, as we know, many bacteria, including Staph aureus, have developed resistance against penicillin. So what they had to do is they had another antibiotic to fight off those resistant strains. And the antibiotic that they developed was a modified version of penicillin called methicillin. All right. And they used methicillin to treat these bacteria here. And it worked for years. But then through overuse of methicillin, we developed these strains of resistant bacteria that we call the MRSA. MRSA, I'm sure you've heard of MRSA before. MRSA stands for methicillin-resistant Staph aureus. These are Staph aureus bacteria that are resistant against the antibiotic methicillin, so it just doesn't work against them. So they had to, do some research and do some studies to find an antibiotic that would inhibit or be used to treat MRSA infections. And currently, if a person is infected with a MRSA infection, they would be given vancomycin. So vancomycin is one of the antibiotics that can be given to fight off MRSA infections. But here's the problem, is that the more and more that we use vancomycin to treat MRSA infections, what's happening is that now we're seeing more and more bacteria that are resistant against vancomycin. We're starting to see what are called VRSAs, MRSA infections. These are Staph aureus bacteria that are vancomycin resistant Staph aureus, and they are very, very hard to treat. There are some antibiotics that we can use against them, but again, it becomes increasingly problematic as we use these antibiotics more and more. Bacteria, they don't have brains, but they have a really good ability to adapt and evolve ways to live under unfavorable conditions. Okay. All right. Now, we can't really have a discussion about inhibiting or blocking cell wall synthesis without having a discussion about, well, what types of antibiotics are there to treat infections caused by, okay, I'm having this issue with drawing again. What type of antibiotics are there that can fight off acid-fast bacteria? So remember that acid-fast bacteria are any bacteria that have these mycolic acids in their cell walls. And remember that mycolic acids are fats. And there's usually two genuses of bacteria that are in this category, Mycobacterium and the genus Nocardia, which we hardly ever talk about in class. But those are the two genuses of bacteria that are considered to be acid-fast that have these mycolic acids. Well, if a person, so, like Mycobacterium tuberculosis or Mycobacterium leprae, the problem is that those fats, these mycolic acids, they're so thick that it really prevents the entrance of a lot of other antibiotics. So if a person had tuberculosis, you can't just, give them penicillin or something else like that because to cure that infection, because those antibiotics, they just can't penetrate the mycolic acids. They're just so thick, they just no way that they can get in. So what you've got to do is you've got to also administer antibiotics at the same time that you're giving other antibiotics. You've got to also administer antibiotics that can get rid of the mycolic acids. And if you get rid of the mycolic acids, then it's more likely that other antibiotics that have other targets can get in. All right. So there are two antibiotics that are usually given at the same time that what they do is they get rid of the mycolic acids. And the two that I'm referring to are these, Isanazid and ethambutol. So again, their target is the mycolic acids, but they target it in a little bit of a different way. Isanazid, what it does, it inhibits the synthesis of mycolic acids. It basically tells bacteria that they can't make them anymore. And then ethambutol destroys any existing mycolic acids. So if there's already any mycolic acids already incorporated into the cell wall, ethambutol will find them and destroy them. So you give both of these at the same time to ensure that you totally, at least as much as possible, you eliminate any mycolic acids in the cell wall. And if you can do that, then also along with Isanazid and ethambutol, you give other antibiotics that can then penetrate the cell and target other things, okay? And we'll talk about that when we get into the treatment of TB. We'll talk about how there's a lot of combination treatment with that. But almost always, especially like with TB, you give Isanazid and ethambutol along with other drugs at the same time, okay? So these are typically used for acid-fast bacteria. All right. So far, what we've done is we've talked about antibiotics that target the cell wall. We talked about beta-lactam drugs. We talked about non-beta-lactam drugs, and then we talked about these antibiotics that inhibit mycolic acids. They're generally used for those acid-fast bacteria. So now we want to move on to my next mode of action, which is to target the cell membrane. And there aren't a lot of drugs that I want to talk about in this category here. There's only one, polymyxin B. So now, if you have a tube of Neosporin or something equivalent to that, if you read the label, you'll notice that there's 2 antibiotics in Neosporin. One of them we just talked about, which is bacitracin. The other one is polymyxin B. And what polymyxin B does, it pokes holes into the cell membrane of bacteria. So when you put Neosporin on your skin, you're using a combination therapy. You're using bacitracin, which inhibits the cell wall, and at the same time, you're using polymyxin B that pokes holes in the cell membrane. All right. And I want to write this up here. Combination therapy is something that we're going to come back to again, because in combination therapy, you're using 2 or more antibiotics that target different things. And that's a good thing, because if you're using one antibiotic that targets one target, then you can't really be 100% sure that you're going to totally cure that infection. So by using 2 or 3 antibiotics that target different things, it increases the likelihood that you kill the infection. Okay, so combination therapy is a very, very common practice, and this is an example of that. Okay. All right, now we're getting into this fairly big category of antibiotics that target the ribosome. They disrupt translation at the ribosome. And by disrupting translation at the ribosome, essentially what this is saying here is that they are blocking protein synthesis. They are basically inhibiting protein synthesis. And the way that they're doing this is by, again, attacking the ribosome. Now, let's kind of refresh our memories on the structure of bacterial ribosomes. So if we look at a bacteria's ribosome, it would kind of look like this here. So remember that a bacterial cell's ribosome has a 50S large subunit combined with a 30S small subunit. Okay. And these antibiotics that we're going to talk about, they either target the 50S large subunit or the 30S small subunit in bacteria. Now, how does the ribosomes of a bacterial cell differ from our ribosomes? What is the structure of our ribosomes? Does anybody remember? Correct. We have 60-40. In our cells, the large subunit is 60 and the small subunit is 40. So this is important. This is an important thing here, because these antibiotics, they would be technically selectively toxic antibiotics. They have a high chemotherapeutic index where they target the 50S subunit or the 30S subunit of bacterial cells, and they do not target the 60 and the 40 of our ribosomes. So because of that, they would be described as being selectively toxic, and they would have a fairly high chemotherapeutic index because they're targeting bacteria and not our cells. All right, now, the other thing before I get into the specific antibiotics that are in this group, in your notes, it tells you, okay, this antibiotic targets the 50S or this antibiotic targets the 30S. And honestly, I really don't care. Okay. What I really care about is that these antibiotics that we're going to talk about, that they target the ribosome, and by doing that, they prevent protein synthesis. But I am not going to ask you which antibiotic targets the 50S subunit or which antibiotic targets the 30. Just know that it attacks the ribosome, and it attacks the prokaryotic ribosomes. And that's really all I'm worried about. Okay. So let's talk about the first group, the first antibiotic in this group, the aminoglycosides. Okay. So the aminoglycosides, so again, they target the ribosome, bacterial ribosomes, and by doing so, they prevent protein synthesis. There's nothing specific that I can say that it's used against, because aminoglycosides, although they're not really broad spectrum, they can be used against gram negatives and gram positives. So they can be used against a lot of different things. But the aminoglycosides do have a pretty serious side effect if they're used extensively. And one of those side effects is ototoxicity, or more commonly known as hearing loss. Another side effect for using them too long is nephrotoxicity, which is kidney damage. Okay. So for the aminoglycosides, know that they inhibit protein synthesis, okay, and that through prolonged use, it can cause hearing loss or kidney damage, okay? And here are some examples of aminoglycosides. Some of these you may have heard of before. So like gentamicin is an example of an aminoglycoside. Tobramycin, which is typically used it can be used to treat eye infections and also external auditory canal infections. Another example of that. Again, you don't have to worry about knowing those brand names, but they are aminoglycosides. All right, our next protein synthesis inhibitor are the macrolides, or better known as erythromycin. Okay, so again, they target the ribosome, and thereby doing so, they inhibit protein synthesis. Now, erythromycin, again, it can be used against a lot of different things, but I'm going to narrow it down for you. Okay, so this is an example of where we have an antibiotic that I'm going to tell you is used against a specific group of organisms. So erythromycin is typically used to treat walking pneumonia, which is caused by Mycoplasma pneumoniae, or Legionella pneumophila infections. Legionella pneumophila causes a disease that we've kind of mentioned before called Legionnaire's disease. Okay, so if anyone has walking pneumonia or Legionnaire's disease, the antibiotic of choice is erythromycin. All right. Not a lot of toxicity to talk about with erythromycin, but that's what it's typically used against. All right, our next one here, chloramphenicol. So chloramphenicol, again, inhibits the ribosome, inhibits protein synthesis by doing that. And this antibiotic, one interesting thing about this, well, a couple of interesting things, is that it is broad spectrum. So it can kill a wide variety of gram positives and a wide variety of gram negatives. And one other important fact about chloramphenicol is that it can cross the blood-brain barrier. And one of its uses is to treat meningitis. So if a person had a really severe form of meningitis, chloramphenicol can be used to treat it, okay, because it can cross the blood-brain barrier and get into the meninges and the cerebrospinal fluid. But here's the problem. The only time that chloramphenicol would be used is as a last resort. You know, what I mean by that is, let's say that all of these other antibiotics that they've tried to treat this person's meningitis, they haven't worked. If that's the case and the patient's going to die and we need to save the patient, then they might use chloramphenicol. But they'll try to hold off using chloramphenicol as much as possible because chloramphenicol has a pretty severe side effect called aplastic anemia. This antibiotic can suppress bone marrow formation. Now, if you remember, bone marrow has got a lot of different functions, and one of those functions of bone marrow is to produce red blood cells and white blood cells. So it is possible that chloramphenicol can destroy a person's bone marrow, and that could cause a lack of an ability to produce red blood cells and a lack of an ability to produce white blood cells into this condition called aplastic anemia. So that's why it's not given as like a general antibiotic. It's usually given as a last resort where, again, this patient has got meningitis. It's a late stage meningitis. We've given all these other antibiotics and nothing seems to be working. Then, okay, well, we might then give chloramphenicol, but this is a different side effect there. Okay. Our next one in this group, clindamycin. So it's another protein synthesis inhibitor that, again, I hate to sound like a broken record, but it attacks the ribosome, thereby blocking protein synthesis. Clindamycin is a very broad spectrum antibiotic, okay? And therein lies the problem. So Clindamycin is usually given to a person if they have like an infection in their tissues or in some sort of body system where, again, the doctor doesn't know what the person's infected with. They might give the person clindamycin because the infection seems to be spreading in their tissues and spreading to other organs. So to cover all the different possibilities, they might give the person clindamycin. So on the one hand, that's a good thing, but it's a bad thing because it's so broad spectrum that it can kill the normal flora in the gut, mainly in the large intestine. Now, if that happens, okay, if a person is on clindamycin and it kills most of the bacteria in their large intestine, then what happens is that this bacteria, Clostridium difficile, will move into the large intestine and cause an infection there. Now, normally Clostridium difficile cannot grow very well in the large intestine, okay, because if you've got a healthy normal flora colonization, the normal flora there prevents Clostridium difficile from moving in. But if you eliminate your normal flora, then there's nothing to stop Clostridium difficile from moving in and causing infection. And if that happens, this bacteria, it'll set up shop in your large intestine and it'll produce a toxin that pretty much destroys the intestinal lining. And that leads to a disease called pseudomembranous colitis. And people that experience this, they have a lot of diarrhea, cramping, bleeding in the stool. Clinically, for those of you that might work in a clinical situation or a hospital situation, they don't actually say the name of the bacteria. They refer to this as C. diff. So if you've ever heard of C. diff, that's what this is. It's an infection in the large intestine caused by Clostridium difficile, and it leads to this pseudomembranous colitis condition. And now I've never had any first-hand experience of this, but in people that have this growing inside of them, not only are they experiencing a lot of diarrhea and all this other stuff, but there's also a very foul odor associated with it. It's a really horrific odor that's really unique to the fact that this bacteria is growing in the large intestine. Okay, so this is kind of a consequence of being given clindamycin for a long period of time. This is a colonoscopic view of a person that has a Clostridium difficile infection in the colon. And what I want to point your attention to is these little white spots that you see here. Those normally shouldn't be there. And this is actually in the beginning stages of this. If this was a really severe case, those white spots would be really big and kind of cover the inside surface of the intestines here. But this can cause, especially you can see a lot of this yellowing right here. That's also a sign of this infection. But this can cause a lot of pain, a lot of discomfort, diarrhea, and even bleeding in the stool. Tetracyclines. Tetracyclines, again, target bacterial ribosomes and prevent protein synthesis by doing so. Tetracyclines are also fairly broad spectrum. And because they're broad spectrum, one of the consequences of being given tetracycline is kind of the same with clindamycin, that it can kill the normal flora and cause infections from other bacteria. And even though it can be used against a lot of different things, I'm going to try to make it easy for you because there is one specific thing that tetracycline can be used for. It can be used to treat acne, especially really, really severe acne. What I mean is like if a person has acne and they've tried a lot of other things like creams and things like that, and nothing seems to work, then it's possible that they might be given tetracycline to treat it. One of the main causes of acne is this bacterium called Propionibacterium acnes. Infects the skin. The only problem is that tetracycline has some side effects. One of the ones is that it's so broad spectrum that it can kill the normal flora. But the other one is that it can impair bone growth. Okay, it can actually impair bone growth. So for one, because of this, you do not want to give tetracycline to a child. Because again, children are still growing, they're still making bones. So if you give a child tetracycline, it can inhibit their bone growth. So you primarily give it to adults that have severe acne. And even in adults, there's a side effect that can happen. It can get into calcium matrix in your teeth and cause discolored teeth, like this. So this person here, they were probably taking tetracyclines for a long time, and the tetracycline got into the calcium matrix of the teeth and caused this discoloration. There's nothing that can cure this cosmetic effect that you see. There's no sort of bleaching technique that a dentist can do to get rid of this. Typically, if a person is experiencing this, they either have to live with it, or if they really want to cover it up, they use porcelain veneers to cover it up, just to cover it up. But there, you can't get rid of that discoloration. Now, remember that I talked about you would rather have acne. Well, it kind of depends on the type of acne, I guess, right? I guess that's, kind of a double-edged sword right there, right? So anyway, so remember that I talked about how there's some bacteria that are resistant against vancomycin. We talked about VRSAs, which are the vancomycin-resistant Staph aureus, but there's also VREs, which are vancomycin-resistant Enterobacteriae. Enterobacteria, these are bacteria that live in your large intestine, like, I don't know, Salmonella, E. coli. So there are certain types of these types of bacteria that are resistant against vancomycin that we need to have antibiotics to treat for. And again, your possibilities are limited, but there are some exceptions to this. There's a group of antibiotics that are referred to as the streptogrammins. And these are some fairly new antibiotics that are being used to treat either VRSA infections or Vera infections, vancomycin-resistant Enterobacter infections. And these are also protein synthesis inhibitors. They target the ribosome. Another example of this, the oxazolidinones. One thing about the oxazolidinones is that they're totally synthetic. They're made by some people in a lab. They work by inhibiting protein synthesis, but these are also fairly new drugs that are being used to treat vancomycin-resistant bacteria. All right. Okay, so that takes care of that fairly broad group of antibiotics that inhibit bacterial ribosomes. All right. So now we're going to get into some antibiotics that fall into this next mode of action, interference with a metabolic pathway. Now, when it says metabolism here, we're not really talking about interference with ATP production. We're talking about interfering with a biological pathway that's typically used to make some sort of product. Okay, so I'm going to diagram something out here first. Bring up a whiteboard. Get rid of this. So let's say that I'm a bacterial cell and I have to make some sort of final product. I'm not going to define what this final product is yet, but for now, I'm just going to call this final product D. We're just going to call it some random final product D. So in order to make D, I've got to start off with some chemical or substrate A. So somehow I've got to get from A to D, but I can't get there in one step. I've got to get there in multiple steps. And the first step in going from A to D is to take A, whatever this may be, this chemical A, and convert it into some chemical B. And then once I've got this chemical B, then I take this chemical B and I convert it into this chemical C. And then I convert C into D, and now I've got my final product. So in order to make this final product, I had to go through 3 steps: A to B, B to C, C to D. All right, so this is a typical metabolic pathway, a biological pathway. Very, very common in a lot of cells. Now, each step in this pathway here, going from A to B, B to C, and C to D, each step is regulated by its own enzyme. So in other words, in order to convert A to B, I had to use an enzyme to do that. Now, I'm just going to call this enzyme. Let's just call this enzyme number 1. So enzyme 1 was used to convert A into B. All right, and once I converted A into B, then I had to convert B into C. Well, can I use enzyme 1 to convert B into C? No, I can't. Okay, to convert B into C, I have to use a different enzyme because, enzymes are proteins that catalyze biological reactions, and one of the characteristics of enzymes is that they can only catalyze one specific reaction at a time. So enzyme number 1, the only thing that it can ever do is convert A to B, A to B. To convert B into C, I would need a different enzyme. So let's say, okay, I'm going to put in here enzyme number 2. So enzyme 2's job is to convert B into C. And then once I make C, then I have to convert C into D. Well, I can't enzyme 1 or enzyme 2. I need a totally different enzyme whose specific job is to convert C into D. So there you go, enzyme number 3. So enzyme 3's job is to convert C into D. So let's kind of think about, well, what is it about an enzyme? Or can anybody tell me what is it about an enzyme that makes it only specific for catalyzing one specific reaction? Does anybody remember from their previous classes? No, I'm talking about what is it about enzyme? Okay, it's specific, but why is it specific? Okay, so it works like a lock and key. So what we're trying to get at here is that enzymes have a very specific shape that can only allow them to catalyze one specific reaction. It goes back to that whole structure and function thing. A particular structure dictates one function. Well, enzymes are like that. They are proteins that have a very specific shape or structure that only allows them to have one function. So enzyme number 1, it has a very specific shape that only allows it to catalyze the reaction converting A into B. Well, okay, let me get into this here. We're going to get into a little what's called enzyme kinetics here, which gets us a little into biochemistry a little bit. But let's say that substrate A, okay, this starting material here, A, is shaped like this. It's kind of shaped like a square. Well, what that means is that enzyme 1 has to have a certain structure to it, a shape to it that can accommodate the binding of A. Now, I'm going to do something here. So all enzymes, they have a little pocket that is typically called an active site or a binding pocket. But what that active site is, it's basically where the substrate has to bind to. And in order for the substrate to bind, the substrate has got to have the correct shape in order to fit into the active site. That also can have it has to have the shape to accommodate it. So if substrate A is, in my example here, if it's shaped like a square, then the active site for enzyme 1 has to have that kind of same shape to accommodate. So it's like a lock and key, as many of you said. The substrate A is the key, and the active site is the lock. And so if the key can fit into the lock, then you open the door here, or you activate the enzyme. So what'll happen is that substrate A fits into the active site, enzyme number 1, and then enzyme 1 does its magic and converts A into B, which now B is a totally different chemical. And let's say that B is shaped like a circle. All right, well, can enzyme 1 be used to convert B into C? No, because enzyme 1 has got more of a squared off active site, and substrate B is more of a circle. So what would the active site of enzyme 2 have to look like to accommodate for the binding of B? It's going to have a circular active site, exactly. So enzyme 2 here, if we kind of draw a basic example of this, it's going to have to kind of look like this. So then B will fit into here, and it'll bind to that little pocket, and then enzyme 2 will do its magic and convert B into C. And let's just say, I don't know, I'm running out of shapes here, so we've got a triangle here. Okay, so that means that enzyme 3 is going to have a binding pocket that can accommodate for C to bind. Okay, and then finally, enzyme 3 will convert C into D. All right. Why am I talking about all this in reference to this? So the drug that I'm going to talk about next here is called sulfonamide. And sulfonamide is a drug that works as a competitive inhibitor. Oops, competitive, sorry. It works like a competitive inhibitor. All right. So sulfonamide is a competitive inhibitor for the production of something called folic acid. So I'm going to replace here as my final product, I'm going to replace D. All right, we're going to replace this with folic acid. Now, let's kind of stop and think here for a minute. So bacteria, they have the ability to make their own folic acid, and they use different enzymes to do that. You and me, where do we get our folic acids from? Do we make our own folic acid? No, we don't. Okay. So where do we get our folic acid from? Yeah, we eat it. We eat it from things that we eat. So yeah, you can get it from vitamins, but you also get it from things like nuts and grains and things like that. You get it from your food. So bacteria, they have these enzymes that allow them to make their own folic acid. You and me, we don't have any of these enzymes. Okay. So sulfonamide is a pretty selectively toxic drug because it only attacks the enzymes that bacteria have. We don't have these enzymes. And sulfonamide, it is a competitive inhibitor. Oops, sorry. Give me a second. So sulfonamide is a competitive inhibitor of the first enzyme. It actually blocks that first enzyme in the pathway of making folic acid. And the way that it does that, hold on. The way that it does that is it acts like a competitive inhibitor of it. So what I'm saying here is that in order to block this enzyme right here, the sulfonamide drug has a very similar shape or structure to our substrate here. So if our substrate A is shaped like a square, the sulfa drug is also shaped like a square. Okay. And the idea here is that if the sulfa drug binds into the active site of enzyme 1 first, okay, what the sulfa drug is doing, it's competing with the substrate for binding into the active site. And if it gets there first, it blocks the active site. And if that happens, will you ever get A converted into B? No. And if you don't get B, you can't get C. And if you can't get C, you can't get folic acid. All right. So that's what we mean when we say that an antibiotic is a competitive inhibitor of an enzyme. It's something that looks very similar to the substrate, and because of that, it can bind to the active site, block it, and prevent the actual substrate from binding in. Okay. All right. So let me show you what's going on here. So let me go back to my slide. Give me a second. Hold on one second here. All right. So in order to make folic acid, this is the reason I drew this example as a three-step process. It is a three-step process. What bacteria have to do is they do have to go through a three-step process where they convert some precursor 1 into precursor 2, and they use enzyme 1 to do that. And then they create convert this precursor 2 into something called dihydrofolate, and they use enzyme 2 to do that. Okay, hold on one second here. Let me get through this and I'll answer the question. And then dihydrofolate gets converted into tetrahydrofolate, which is actually your folic acid. So you actually have 3 enzymes to do this. And these sulfa drugs, they block the first enzyme. Now, in my example that I drew on the whiteboard a little while ago, I said that the substrate of the first enzyme was something called A. Well, it's not actually A, it's actually something called PABA, or para-aminobenzoic acid, or PABA. And so the sulfa drug, the sulfonamide, competes with PABA for binding into enzyme 1. And let's take a look at the structure between PABA and sulfonamide. Well, here it is. Here's on the one on the left, you have PABA, and on the right, you have sulfonamide. And you can see that with the exception of a few chemical groups here, they almost look identical. All right. So sulfonamide, if it binds to enzyme 1 first, it blocks the ability of PABA from binding. And if you do that, you basically inhibit this process. You inhibit the production of folic acid synthesis right here at that first step. Okay, right there. Okay. Now, the only problem here is that a lot of times when sulfonamide is being given to treat some sort of infection, you don't give it by itself. Okay, because what if some of the bacteria that you're trying to kill, what if they already made it past that first step? So they've already made it to here. Well, if they've already done that, then they could still use enzyme 2 and enzyme 3 to make folic acid. So typically, when you're giving sulfa drugs to prevent folic acid synthesis, you don't give it by itself. You give another drug called trimethoprim. And what trimethoprim does, it blocks the third enzyme. So you block the first enzyme and the third enzyme to help to ensure that you prevent folic acid synthesis. A very common antibiotic that has sulfonamide and trimethoprim in it is Bactrim. And one of the more common uses for this is to treat UTIs, which are urinary tract infections. I'm not saying that that's the only thing it's used for, but it's one of the things it's used for. Okay, Elsie, you had a question here. You had your hand up. Oh, it was by accident. Okay. All right, so I think we'll stop here. I don't have enough time to talk about our next mode of action here, which talks about inhibition of nucleic acid synthesis. So we'll pick up with that, and then we'll get into talking about some other. So I will see you on Wednesday, and if I don't talk to you then, have a good couple of days. All right, take care. Thank you very much.